Dear students, Assalamu alaikum. This is Dr. Sohail Aslam. Today, we will be starting the study of the male reproductive system. Remember, even though the reproductive system does not contribute to homeostasis and is not essential for survival of an individual, it still plays an important role in a person's life. It has important influences on how people view themselves and how they interact with others. Reproductive functions also have very profound effects on the society. This lecture consists of following sections: basic concepts, embryology, aberrant sexual differentiation, and physiological anatomy if we are left with any time. It will be quite appropriate here to go through some basic concepts regarding male reproduction. The multiple differences between the male and the female depend primarily on a single chromosome, that is the Y chromosome, and a single pair of endocrine structures, which are the testes in the male and the ovaries in the female. The differentiation of the primitive gonads into testes or ovaries in utero is genetically determined in humans, but the formation of male genitalia depends on the presence of a functional secreting testes. Therefore, in the absence of testicular tissue, development is female. Okay, I will now review some important points about sex differentiation and development. We, from our previous knowledge, know that sex is determined genetically by two chromosomes called the sex chromosomes, to distinguish them from the somatic chromosomes, the autosomes. In humans and many other mammals, the sex chromosomes are called X and Y chromosome. The Y chromosome is necessary for the production of testes. The testis determining gene product is called SRY. SRY stands for the sex determining region of the Y chromosome. SRY protein acts as a transcription factor that initiates transcription of number 1, genes necessary for testicular differentiation, and number 2, gene for Mullerian inhibiting substance. We know that there are 46 chromosomes. In males, there are 22 pairs of autosomes and 1 pair of sex, that is X and Y chromosomes. In females, there are 22 pairs of autosomes and 1 pair of sex, that is XX chromosomes. This figure shows the basis of genetic sex determination. In the two-stage meiotic division, in the female, only one cell survives as the mature ovum. In the male, the meiotic division results in the formation of 4 sperms, 2 containing the X and 2 the Y chromosomes. Fertilization, therefore, can produce a male zygote with 22 pairs of autosomes plus an X and a Y chromosome, or a female zygote with 22 pairs of autosomes and 2 X chromosomes. Human chromosomes can be studied in detail. Human cells are grown in tissue culture, treated with the drug colchicine, which arrests mitosis at the metaphase, exposed to a hypotonic solution that makes the chromosomes swell and disperse, and then squashed onto slides. Staining techniques make it possible to identify the individual chromosomes and study them. The individual chromosomes are arranged in a pattern called karyotype. This figure shows the karyotype of chromosomes from a normal male. Let's now study inactivation of X chromosomes. Soon after cell division has started during embryonic development, one of the 2 X chromosomes of the somatic cells in normal females becomes functionally inactive. The choice of which X chromosome remains active is random. Consequently, some of the somatic cells in adult females contain an X chromosome of paternal origin, and some contain an active X chromosome of maternal origin. In normal cells, the inactive X chromosome condenses and can be seen near the nuclear membrane as the Bohr body, also called sex chromatin, and it's also present as a drumstick in polymorphonuclear leukocytes. This figure shows cells of the spinous layer of epidermis. The left panel of the figure shows cells of a female, and Bohr bodies are shown pointed by the arrow. The right panel shows cells of a male, and you can easily make out that there is no condensation of X chromosome near nuclear membrane, and hence no Bohr bodies are present. This figure shows nuclear appendages in white blood cells. The left panel shows white blood cell of a female, and you can easily see the drumstick identified by the arrow. However, the right panel shows blood cells of a male. There is no drumstick present in the cell in the center of the figure. Next section of the lecture deals with the embryology of the human reproductive system. This is important to study because it helps us to understand various abnormalities of sexual development. The development of the gonads. On each side of the embryo, a primitive gonad arises from the genital ridge, which is a condensation of tissue near the adrenal gland. The gonad develops a cortex and a medulla. Until the 6th week of development, these structures are identical in both sexes. However, in genetic males, the medulla develops during the 7th and 8th week into a testes, and the cortex regresses. Leydig and Sertoli cells appear. Testosterone and Mullerian inhibiting substance is secreted. In genetic females, the cortex remains and develops into an ovary, and the medulla regresses. The embryonic ovary does not secrete hormones. In the 7th week of gestation, the embryo has both male and female primordial genital ducts. Considering internal genitalia, in a female fetus, the Mullerian duct system develops into uterine tubes and a uterus. In the male fetus, the Wolffian duct system on each side develops into the epididymis and vas deferens. The external genitalia are similarly bipotential until the 8th week. After that, the urogenital slit disappears and male genitalia form, or alternatively, it remains open and female genitalia form. When the embryo has functional testes, male internal and external genitalia develop. The Leydig cells of the fetal testes secrete testosterone, and the Sertoli cells secrete Mullerian inhibiting substance. In their effects on the internal as opposed to the external genitalia, testosterone and Mullerian inhibiting substance act unilaterally. Testosterone promotes the development of vas deferens and related structures from the Wolffian ducts. The enzyme 5-alpha reductase changes testosterone to its active metabolite dihydrotestosterone, which induces the formation of male external genitalia and male secondary sex characteristics. On the other hand, Mullerian inhibiting substance causes regression of the Mullerian ducts by apoptosis on the side on which it is secreted. Therefore, no female internal genitalia can develop in a normal male fetus due to actions of Mullerian inhibiting substance. I will pause here a while to let you have a good look at this figure. This figure shows the diagrammatic summary of normal sex determination, differentiation, and development in humans. Okay, we now move on to the next section, which is aberrant sexual differentiation. In this section, we shall study various disorders which can result due to abnormal sexual development. Abnormalities of abnormal sexual development can be classified into two major types: chromosomal abnormalities and hormonal abnormalities. Let's first study the chromosomal abnormalities. Nondisjunction. By definition, nondisjunction is a phenomena in which a pair of chromosomes fail to separate during meiosis, and therefore both go to one of the daughter cells. Non-disjunction of sex chromosomes during the first division in meiosis results in distinct defects. Although non-disjunction usually occurs during the first meiotic division, it can occur in the second, producing more complex chromosomal abnormalities. transposition. Chromosomal abnormalities also include transposition of parts of chromosomes to other chromosomes. Rarely, genetic males are found to have the XX karyotype because the short arm of their father's Y chromosome was transposed to their father's X chromosome during meiosis, and they received that X chromosome along with their mothers. Similarly, deletion of the small portion of the Y chromosome containing SRY gene produces females with the XY karyotype. Okay, now this is a very good figure to understand the various chromosomal abnormalities. This figure shows the summary of 4 possible defects produced by maternal non-disjunction of the sex chromosomes at the time of meiosis. For your convenience, I have split this diagram into 4 parts so that we can study the 4 defects one by one. This figure shows that the ovum did not receive the X chromosome from the mother. Therefore, the fertilized zygote has the 44XO chromosomal pattern. O means 0. This results in gonadal dysgenesis, which is also known as Turner syndrome. In individuals with the XO chromosomal pattern, the gonads are rudimentary or absent. Female external genitalia develop. The stature is short. No sexual maturation occurs at puberty. This figure shows that the ovum received both X chromosomes from the mother. Therefore, the fertilized zygote has the 44XXY chromosomal pattern. This results in seminiferous tubule dysgenesis, which is also known as Klinefelter syndrome. The individuals with the XXY pattern, this is the most common sex chromosome disorder. They are normal male genitalia and male characteristics. However, mental retardation may be present in some of the individuals. This figure shows that the ovum received both X chromosomes from the mother, and the fertilized zygote has the 44XXX chromosomal pattern. Individuals with this pattern are also known as superfemales. This is the second in frequency only to the XXY pattern, and it is more common in the general population and not associated with any characteristic abnormalities. This figure shows that the ovum did not receive the X chromosome from the mother, and the fertilized zygote has the 44YO chromosomal pattern. O means 0, and this is not compatible with life. The YO combination is believed to be lethal. The fetus dies in utero. Having discussed the disorders resulting from chromosomal abnormalities, I will now describe the disorders that result from hormonal abnormalities. The term pseudohermaphroditism comprises the major disorders that occur because of hormonal abnormalities. A pseudohermaphrodite is an individual with genetic constitution or gonads of one sex and external genitalia of the other sex. A pseudohermaphrodite may be a male or a female. Male genital development can occur in genetic females exposed to androgens from other sources. The causes of female pseudohermaphroditism, therefore, may include congenital adrenal hyperplasia or virilizing tumors, or it may be caused by androgens administered to the mother. Male pseudohermaphroditism. The male pseudohermaphroditism may result from defective testicular development. As the testes also secrete Mullerian inhibiting substance, these genetic males have both external and internal female genitalia. Another cause of male pseudohermaphroditism is androgen resistance, in which male hormones cannot exert their full effects due to various congenital abnormalities. There may be deficiency of 5-alpha reductase, which converts testosterone to its more active form, dihydrotestosterone. Other forms of androgen resistance are due to various mutations in the androgen receptor gene. Testicular feminizing syndrome or complete androgen resistance syndrome results when the loss of receptor function is complete. This is another form of male pseudohermaphroditism. In testicular feminizing syndrome, what happens is that Mullerian inhibiting substance is present, therefore no female internal genitalia develop, and testosterone is secreted at normal rates but cannot act on the target tissues. Therefore, the external genitalia are also female. Now, individuals with this syndrome develop enlarged breasts at puberty and are considered to be normal women until they seek medical advice because of lack of menstruation. This concludes today's lecture, and you have to answer this quiz in your responses when you mark your attendance.